So I wanted to start by a story about glycation. Uh, and the story involves my children, actually. I have two daughters, 5 and 9. And one morning, they refused to eat breakfast. They just said that they're not hungry. And as a parent, you're worried what's happening, and how can you give them food. So I happen to have a air fryer I recently bought, and I decided to make some fries. So I took potatoes and you fry them in that. And suddenly after I made those, they ate fries for about 1 hour. They were able to eat about 10 potatoes. So I was shocked, and I was like, what's going on? They wouldn't eat their normal breakfast, but they would eat these fries. Why is that? So I mean, just showing you kind of what happened at our home. So what I want to tell you about today is something called advanced glycation end products. So the act of making fries actually creates what's called advanced glycation end products. So basically, for example, bread, when you toast it, basically if you have proteins and glucose and you provide heat, especially around about 370 Fahrenheit, you within 4 or 5 minutes, you change the flavor. And this browning is called, this is described as a Maillard reaction. This was discovered in 1900s, and actually since then has been used a lot by the food industry, and it's also used to create meat flavor. So a lot of people don't know about glycation, but this is the most commonly practiced chemical reaction. Every time you're cooking, just imagine how much you could eat if you don't cook. Basically, it's been seen in many studies, if you just eat raw food, you just can't take the same amount of calories. So why am I telling you all this? I think this is in part responsible for the obesity epidemic we are seeing all around the world. So despite all our understanding, which is improving every year, the obesity trends are going in the wrong direction. Why is it that despite increasing knowledge and medicines, we're actually, the levels of obesity are rising all across the globe? Why is obesity a problem? We know that it is a risk factor for so many diseases, and especially for today's talk, some of the relevant diseases are diabetes, cardiovascular diseases, Alzheimer's disease, and dementia. These are some of the biggest killers, and these are all, in part, obesity contributes to accelerate them. Now, none of you may be surprised by this. If you think how much we are surrounded by these foods that are glycated and especially full of sugars, this is not surprising that we are now eating almost 10 to 50 times more sugar per capita than we were eating about 100 years ago. I mean, this is a shocking statistic if you think about it, but it's not so shocking if you think about it in the light of evolution. So if you think of how we evolved, I think, I don't blame anybody who overeats, by the way. I myself have a sweet tooth. I gravitate towards sugary food, and one can see why. The reason, one of the ideas is that during evolution, this was actually a beneficial trait. When food was scarce, people and organisms who can take in extra food actually are, they do better if you think in terms of obese, like when food is scarce, right? Under conditions of starvation, they would actually do better. So food addiction even had a survival value in our past. However, the problem is not the genes for obesity and diabetes. The problem is the modern situation we are in. We're constantly surrounded by food, and our food has become a lot more addictive than it ever was, which is creating this problem. So we have done an amazing job to create, wonderful flavors, but the problem is we don't know when to stop. And the question we are asking in my lab is carbohydrate toxicity avoidable, and is it safe to turn it down, right? So I'm a faculty at the Buck Institute, and I just want to also declare a conflict of interest that based on the work I'm showing, I've also started a company called Juify Health, and one of the things I'm going to talk about, compounds that can help reduce this carbohydrate toxicity and satiety, is called GLYLO, for glycation lowering, and I'll be describing that, and that's also commercialized by the company. So what are advanced glycation end products? So our hypothesis is that it's not sugar that really drives the problems. Sugar is actually not a very reactive molecule, but when sugar is metabolized by cells, it's producing toxic metabolites. So this is something called methylglyoxal. This is a very reactive molecule. It binds proteins, DNA, lipids, and the problem is it reacts with things to cause advanced glycation end products. Now, like I said, all of us know about advanced glycation end products because every time you're cooking, the browning of the bread, the caramelization of onions, all that is advanced glycation end products. Unfortunately, this is happening slowly in our body as well. And we think it's not sugar, but it's advanced glycation end products that is the driver of many diseases related to diabetes and also aging. So for example, diabetes is the leading cause for diabetic retinopathy, nephropathy, neuropathy, cardiovascular diseases, stroke, and even the risk of dementia is 2 to 4-fold with diabetes. And we think one of the major reasons for this is the production of advanced glycation end products. In all these situations, there's been shown to be an increase in advanced glycation end products in our body with age, and also when you have type 2 diabetes especially. But also under conditions of even hyperglycemia, you see this increase in advanced glycation end products. So, and we will review on this if you want to read more about it. And it's also on our website. There's a lot of knowledge about advanced glycation end products if you want to learn more. So just to go in more detail, every cell that uses glucose, this is from bacteria to us, they make these advanced glycation end products. It leaks, and it's called a non-enzymatic reaction because it is happening spontaneously, but there are enzymes in place to stop it. So they're called glyoxylases. Now but it's not, they can't protect you, the proteins get modified. And that's why also the collagen gets modified. So even the skin, in exposure to sunlight, that's also formation of these advanced glycation end products. So it's happening under a number of situations in our body. But as I said, it's happening in our bodies slowly. We do it all the time while cooking, but it's something not very well studied when it happens within the body, but it's a big problem. So we previously showed that actually this is sensed by a receptor. So it's called TRPA1. Recently, the Nobel Prize was given for this. This is also the pain receptor. Interestingly, it senses this precursor for AGEs. And when it senses that, it tells the cell to detoxify it. And this is very important sort of homeostatic mechanism in our body. Every time the body is sensing AGEs, there's a system in place to tell it to detoxify it. Now, what we discovered was that we can use drugs to switch on this system to increase the detoxification. Now, these enzymes involved in detoxification are already associated with Parkinson's disease and diabetic complications. So we know that these enzymes are important. And the important thing is, though, we can use drugs to activate them. And I'm not going to go into the details of how we came up with these compounds, but we did a screen for over 1,000 compounds, and we asked which compounds would activate and protect the cells from damage due to advanced glycation end products, methylglyoxal specifically. And that resulted in a very interesting result where we found that 5 compounds together, nicotinamide, alpha-lipoic acid, piperine, a component of black pepper, vitamin B6 pyridoxine, and thiamine, which is vitamin B1, these 5 components, when we added them together, we had the strongest protection in neurons against methylglyoxal, against the damage due to advanced glycation end products. So basically the idea was, like I said, cells are making these damaging products, and with age and in certain conditions, this is too much and the cell starts aging more rapidly. Now, the question is, can we use compounds to slow down this aging due to advanced glycation end products? So we created this concoction. So I'm going to show you some results which seem very promising with this cocktail of compounds. So we use these 5 compounds. We call them GLYLO for glycation-lowering, right? So I'm going to use this term GLYLO throughout my talk. And like I said, the product is sold by the company as GLYLO, GLYLO. So I don't want anybody to be confused. So first of all, we measured the methylglyoxal, the precursor of these AGEs. And you can see when you give the mice these compounds, within a few weeks, we can see a reduction in these in the level of methylglyoxal. And this is quite important because this has not been done before. It's not easy to reduce the levels of methylglyoxal. And also I want to add that reduction of this, that methylglyoxal is present in our serum all the time. It's there in cerebrospinal fluid. It's there in all the tissues. So if there is a way to lower endogenous methylglyoxal level, we think that would be an advantage, and we've been able to do this. And not only did we reduce methylglyoxal, this simple molecule, like I said, cross-reacts with amino acids, lysines, arginines, and here you can see the cross-link. And we can see that this cross-link is also reduced in the plasma when we give the animals GLYLO. So the next experiment, as we fed these animals, we wanted to know what was going on. And to our surprise, because we weren't initially thinking about appetite, but we found that these animals were eating less. Because initially we were working on methylglyoxal and advanced glycation end products in the context of aging. But like I described to you, because they have such an important role, it looks like from appetite, given how much we use them in our diet all the time, we were not surprised to see that actually it does reduce food consumption. And I will not go into too much details, but we actually found that it reduces specifically carbohydrate consumption. So when we put it on a high-fat diet, we don't see an decrease in the appetite, but on a high-carb diet, they reduce food intake. Secondly, the body weight was reduced. So normally the mice will gain weight. So you can see the mice will start at 30 grams and they become 35 grams in about 4 or 5 months. But when we give them GLYLO, they maintain this body weight. Next, we were encouraged to see these results, and we wanted to know what's happening to levels, glucose homeostasis. So you do something called a glucose tolerance test, where you ask essentially, how well does the animal do to a glucose challenge? So you put in a lot of glucose into the animal, and you measure how long it takes for them to clear. So you can see in black are the control animals. The peak goes up in 30 minutes, and then it's trying to clear it, and it takes about 2 hours. But you can see that with GLYLO, the peak is never the same, and they clear it much faster. So we think these animals, it improves the glucose homeostasis because it can get rid of the sugar much faster. So that means, so if there's any damage due to sugar in the body, that's that will be prevented. Next, we measured insulin tolerance, and we found that these animals are also, their insulin tolerance is also improved. So that means when you give an injection, you give an injection of insulin, these animals can lower their sugar even more. So now the hypothalamus is the part of the body that is important for food intake. So the next question we asked was, what's happening to the brain? How is it that it's reducing this food intake? And we found that, so you're more familiar with the idea of glycolysis. The cells use sugar or also fat or other energy substrates. But the idea in the field is that if you use sugar too much, that's detrimental to the cell. So we've been able to find that if you take GLYLO, essentially, these animals show a reduction in glycolysis. So all the enzymes in the glycolysis pathway, many of the enzymes go blue, except this one, which is good because this enzyme actually helps to reduce the methylglyoxal. So we're able to reduce glycolysis while and we think the production of methylglyoxal. So the amount of methylglyoxal a cell would make now is reduced. Secondly, we find that it's also improving the levels of glutathione. And this is an important antioxidant. So glycolysis is also used to make antioxidants like glutathione. So we find that upon GLYLO treatment, they make more of these glutathione antioxidant. And thirdly, we just looked at different enzymes important for detoxification of methylglyoxal. So like I showed you earlier, there are enzymes in place that are constantly trying to reduce methylglyoxal. And as we expected, our compounds actually increase the levels of these methylglyoxal, the detoxification compounds. Next, we try to get, we asked the question, if this is working in normal mice, how about if we give them to obese mice? So we take mice that are deficient in what's called the leptin receptor, an important gene for maintaining appetite. So when leptin levels are increased, the animal eats less. But if you block this signaling in, the animal gets obese, like you can see here. So when we give the animals these glycation-lowering compounds, you can see here, we call anti-AGEs here, you can see in red, basically these animals reduce weight drastically and they maintain this lower weight. So they actually change the set point, and now they, instead of these animals being about 45 grams or 40 grams, they stay around 30 grams. And more interestingly, we kept these mice in our vivarium for a long time. So after 6 months, half the animals had died because these animals tend to get type 2 diabetes and diabetic complications. But interestingly, the animals kept on the supplement GLYLO essentially were not, none of them died at all. So we think there's some sort of protective effect of glycation-lowering compounds even in this mouse model. So next we asked, why are they eating less? And I'm, again, not going to go through all the data, but there's a hunger hormone called ghrelin. Now, ghrelin is important in telling the animal that it's hungry, and it acts on the hypothalamus, increases AMPK, and TOR, essentially reduces TOR, so to tell the animal that it's time to eat more. However, when we gave the animals GLYLO, we found that AMPK levels were actually not upregulated in the hypothalamus, and the animals were not responsive to ghrelin. And so interestingly, we have a surprising situation here where the animal is like this. So normally on food restriction, TOR will go down and the animal gets hungry. If you just reduce TOR in that part of the brain of the mouse, it would go eat more. However, when we give our compounds, they're actually upregulating TOR somehow, and they maintain satiety. So we're trying to understand why this is happening more, but right now this is what we've sort of understood as to why how these animals are able to control their appetite and reduce their attraction towards sort of sugar-rich diet. Next, we had these animals that which started at the age of 24 months. So we had these old animals, and we asked, what if we were to give them GLYLO? These are normal mice to about 24 months of age, which means they're about 60 to 70-year-old in human age. And when we gave them these compounds, we could see an extension in lifespan. And in this experiment, as you can see, we tested more than 40 animals each on both these different diets. So in black is the control diet, and in red is the diet containing the glycation-lowering compounds, which I described earlier. So just to summarize this, what we are seeing is that glycation-lowering compounds do a number of things. One of them is reducing the signaling in the sort of ghrelin receptor, such that ghrelin is not ghrelin doesn't act to increase food intake when you're hungry, and it also increases insulin sensitivity and extends lifespan. And we also see a decrease in glycolysis, an increase in the pentose phosphate pathway, which increases our antioxidant levels, and lowering of methylglyoxal, overall lowering the AGEs burden, which we think contributes to the aging process. So this is just one story I was telling you, but this has led to a number of exciting projects in the lab, which I don't have time to describe today, but I'll briefly mention that we are seeing a number of other improvements in other tissues in using GLYLO, which is, for example, we're seeing an increase in muscle mass and also improved grip strength and sort of muscle function. There's a slowing of eye aging. There's improvement of cardiovascular function. We can also reverse a number of menopause-related effects, actually. And in a model of Alzheimer's disease, we can also rescue the memory deficit in that model. And also, quite interestingly, in the menopause models, we can reverse the insulin resistance and weight gain that takes place when you take out ovaries from an animal. So I won't get into other times, but essentially also other hallmarks of aging like cellular senescence and mitochondrial function are also improved. So our hope is, like I said, I showed data how we are using C. elegans and neurons to identify these pathways that are important for reducing glycation, and we use these approaches to identify compounds that we tested in mice, and we're now hopeful to take some of our discoveries and test them clinically. And we're looking for partners and clinical partners to work on that. So with that, I want to thank my team, and Lauren Weimer and Kiyomi did most of the work we described here, and my collaborators and my funding sources, AFAR and NIH. And with that, I will be happy to take any questions. Thank you.
Thank you, Dr. Kapahi. So do the on-site audience have any questions for him? Distinguished Kapahi, Professor Kapahi, I have a question. When you were introducing GLYLO, you used the lipoic acid and the papain and other supplements together. Would it be better if I use GLYLO together with other anti-aging drugs such as rapamycin and metformin?
Yeah, that's a great question. So we have not done the human data, so I couldn't answer the human data, but we are doing these experiments in mice. And we combined with 4 different interventions. And it's very preliminary to say right now, but with metformin, there was no further improvement. So I think this overlaps with the effects of metformin. But with rapamycin, there might be some further improvement in like muscle function, grip strength, and their ability to exercise. So we're exploring this more carefully now. But thank you for that question. Yeah. That's something we're very hopeful for. I just want to add something here. In prior research I did, we showed that if you inhibit both the insulin pathway and the TOR pathway in C. elegans, you could extend lifespan in worms. These are small worms. You could extend lifespan by 5-fold. And that was quite dramatic, actually. But these worms only live 20 days, so we could make them live about 100 days. And we are trying to do something similar in mice now. So we think GLYLO can improve insulin sensitivity and reduce IGF. So we're trying to see if rapamycin and GLYLO together would have additive effects on lifespan.
Thank you, Professor Kapahi. Do we have another questions from the floor? So in the live streaming conference room, we also have some questions. I will just choose one. Many audience are curious that for GLYLO, what's the clinical plan of GLYLO? which stage is it now? Do you have any plan for the GLYLO clinically?
So, like, we're working with a number of clinicians right now and writing grant proposals to try small clinical trials around body weight and insulin resistance initially, and also sort of epigenetic markers of aging. So this is in sort of early stages, but we've got approval from the FDA to sort of try the combination, and the next step is to sort of raise funding to try this in humans.
Thank you for your support, Professor Kapahi. Now, I don't want to mess around with foods, highs, and AGEs. Now, looking forward to seeing more of your research.